Hey, micro friends, welcome to another episode of Let's Talk Micro. As always, I'm so happy that you're here with me. And if this is the first time you're listening to Let's Talk Micro, well, thank you for choosing it as one of your podcasts. Okay, so those of you that have been listening for a while, you know that every year after I attend ASM Microbe, I always do a recap episode where I share my favorite talks, posters, things that I saw on the exhibit hall. So this is the episode for the 2026 ASM Microbe recap. So, in case you didn't know, ASM Microbe, which ASM is the American Society for Microbiology. So, Microbe is the largest microbiology meeting in the United States, and it brings together thousands of scientists from all over the world every year. And if this was the first year that you were attending, maybe you didn't notice, but if you have been attending for years, you saw that this year the format changed. In prior years, you had like different tracks, and this year they restructured things. So basically, it was like three meetings in one place. So there was the health meeting, which is the one that I'll be talking about because that's the one that I focus on. And this one is focused on clinical microbiology, such as diagnostics, resistance, infection prevention, basically everything that we do in the clinical lab. And then there was the applied and environmental microbiology meeting, which is about understanding microbial processes across different environments. And then there was the mechanism discovery meeting, which focuses on pathogenesis, host pathogen interactions, and things like that. So, as I said, I will be talking about the health meeting in this episode. And if you want to provide any feedback about the other meetings, please do so via social media, via email, let me know. But one thing that I have to say is that I really enjoyed this structure, the way they did it this year. It felt more organized. In prior years, you had to do a lot of walking to see all the different posters and things like that. And also, you had to do a lot of walking to go to different meetings and different talks. I mean, having said that, I don't know, maybe this venue was more accessible than others. I mean, they all change depending on the convention center where the meeting is held. Sometimes you feel like you have to walk more to get to some places, but in this case everything was more accessible. The posters were very organized, so shout out to ASM. It was a great meeting. And once again, thank you so much for those of you that took the time to attend the live episode of Let's Talk Micro. And this was a huge milestone in the podcast, and I'm happy that I got to see so many of you attended and share this with you. So thank you again. Thank you. Okay, so let's go ahead and start with the talks, and then I'll move on to the posters. So, of course, there's the classic Battle of the Brains. And for those of you that maybe attended the first time or maybe you're not sure what this is, think about it like Jeopardy for microbiology people. And you don't have to give your answers in a question format, but I love the energy, and the questions are very hard. So do not feel bad if you attended this event and you did not know the answer to a lot of the questions. This is supposed to be a learning experience. So when you walk away from that event, you should have learned something. So don't feel bad. And the way that it's organized is very neat. So you get two groups that they compete against each other. Then you get two more groups, they compete against each other. And then the winners from both groups, they compete against each other. And then you have a winner. And typically, the groups can be, for example, like public health microbiologists. Sometimes you see like fellows. And I did participate in 2024, and my group was composed of like microbiology influencers. So it was a great experience. So shout out to this year's winners, which were the fellows. And of course, I mean the CPEP fellows. So, which they're great listeners of the podcast. So shout out to you and congratulations for winning this event. Okay, so moving on to other sessions, one that I attended was called Barking Up the Wrong Tree When a Bug Doesn't Follow the Rules and Why You Still Should. And this was from Robert Bowden from the Beth Israel Deaconess Medical Center. And this was a very neat talk. And he talked about a case where there was this organism that it was ultimately identified as Brucella, but many things went wrong. The biochemicals were conflicting. There was an error with the gram stain, and one of the IDs was Brucella. But because of these errors, the ultimate identification was delayed. I mean, it was eventually sent back to the reference laboratory in the lab response network, but it was delayed. So the takeaway from this, you always have to follow the proper safety procedures when you're working with these organisms. make sure that you understand that they do not read textbooks. So you're going to get abnormal reactions, you're going to get abnormal morphologies. So you have to keep that in mind. But even like in this case, in the lab, there was an idea of Brucella. So if you get something like this, you have to investigate further and follow the proper procedures. Okay, so moving on, another session that I attended that I liked was from Sarah Gerfro from the North Dakota State University Veterinary Diagnostic Laboratory. And she was a guest in the podcast before, but she presented some work on Staphylococcus pseudointermedius in animals. And in this work, they looked over 1,900 Staphylococcus pseudointermedius isolates from dogs and cats and compared individual beta-lactam breakpoints to oxacillin as a surrogate for MEK mediated resistance. And in this work, oxacillin turned out to be the most sensitive and reliable phenotypic marker for MEK, and oxacillin susceptible isolates without MEK were almost always susceptible to the other beta-lactams. In contrast, many individual beta-lactam breakpoints, such as amoxicillin clavulanic acid, cefazolin, and cephalothin, show high rates of very major errors, meaning you could easily call a MEK positive isolate susceptible if you only look at those drugs. That work is feeding into updates in the CLSI veterinary documents, moving away from a long list of individual beta-lactam breakpoints and toward using oxacillin or cefoxytin as surrogates, which is very similar to what we already do on the human side. And you can find this paper in the Journal of Clinical Microbiology. It was published on June 12th. So you can go online and look for it. Very interesting work. And, shout out to Sarah, shout out to Kelly Maddock, and all the microbiologists that are doing great work on the veterinary side. They have a lot of challenges and they're working very hard to make sure that things like breakpoints, they are updated and the animals can get the best possible antimicrobial therapy. Okay, so the next talk that I attended was about escape pathogens and new therapeutics. And this started with Dr. Cesar Arias from Houston Methodist Hospital. And the talk was called Progress in the Development of Antimicrobials Against Escape Pathogens. And he gave a broad overview of the pipeline. On the non traditional side, he highlighted microbiome based therapeutic products for Clostridioides difficile and bacteriophage therapies in development for Pseudomonas aeruginosa, Staphylococcus aureus, and E. coli, including inhaled phage delivery and a trial of phage therapy for Staph aureus bacteremia. And on the small molecule side, he walked through several new beta-lactam beta-lactamase inhibitor combinations, as well as agents targeting the bacterial membrane, protein synthesis, DNA synthesis, topoisomerases, and fatty acid synthesis. The message was that for escape organisms, we're seeing both incremental improvements, such as new beta-lactam beta-lactamase inhibitor pairs, and some new targets and mechanisms, which is encouraging in a space that often feels like it's lagging behind. And then within the same talk, Dr. Pranita Tama from the University of Pennsylvania School of Medicine, she gave a talk titled The Gram-Negative Escape Pathogens, Where Are We Now? And in this talk, she started with the mechanisms we deal every day in Pseudomonas aeruginosa, such as reduced membrane permeability, loss of porins, active efflux pumps, target site modifications, and enzyme-mediated resistance, including Pseudomonas-derived cephalosporinases. And then she walked through how some of the more recent and newer agents, such as ceftazine tazobactam, ceftazine avibactam, uvenarilibactam, cefiderocol, and cefepime-zidibactam, performed against difficult to treat Pseudomonas, including Vim-producing strains. And then she also showed real-world examples where patients develop resistance to agents like ceftazine avibactam or cefiderocol during therapy, and emphasized that for Vim Pseudomonas, our reliable beta-lactam options are essentially limited to cefepime and cefepime-zidibactam, with cefiderocol often used but not always sufficient alone. So this was a good talk on, on how to actually deploy these drugs and where the gaps still are. And then I attended a talk by Pablo Power from the University of Buenos Aires, and it was titled Indelicate Loops, Insertion Deletion Mutations, Rewiring the Resistance Landscape in KPC. And he focused on small insertion and deletion mutations in KPC beta-lactamases and how they change the enzyme's structure and function. And these insertions and deletions can alter substrate specificity and increase resistance to newer beta-lactam beta-lactamase inhibitor combinations, such as ceftazine avibactam. And the big message was that even even once we have a new inhibitor on the market, these enzymes are still evolving under selective pressure. So there needs to be structural work and surveillance for emerging KPC variants, and this is critical if we want our newer beta lactamase inhibitor combinations to stay useful. Okay, and then there was the live episode of Let's Talk Micro, which I'm very grateful for those of you that attended, and we will be releasing it soon. Which was titled "What Can Whole Genome Sequencing Realistically Tell Us About Resistance?" And the episode was co-hosted by Andrew Princey, and in the episode I was joined by Dr. Nathan Littibor from the Medical College of Wisconsin. And it was a great episode. We hear a lot of talks about whole genome sequencing, and this episode was about what that information can provide regarding AST or antimicrobial susceptibility testing. And the question was that can we use this information that we get from sequencing to predict susceptibility testing? So it was a great talk. And as I said earlier, I will be releasing the episode soon. So I hope that you enjoy it when I do. Okay, so those were the talks, and there's so many more, and these are the ones that I enjoyed and they caught my eye. But let's go ahead and move on to the posters. And having said that, I spent a lot of time walking through the posters. I talked to people, recommended the podcast, I took a lot of pictures. So there were some great posters out there. But for the sake of time, I'm going to highlight 6 of them. So let's go. The first one was from Amber Kimball from Howard University, and she presented a poster called Connecting the Dots, Evidence-Based Assessment of the Role of Pharyngeal Gonorrhea and Commensal Neisseria Species in the Emergence of Antimicrobial Resistance of Neisseria Gonorrhea. And basically in the poster, she reviewed how asymptomatic pharyngeal gonorrhea and commensal Neisseria in the oropharynx can exchange resistance determinants and how the pharyngeal microbiome may support persistent infection and antimicrobial resistance, with the goal of improving screening and treatment strategies for pharyngeal Neisseria gonorrhea. The next one was from Matthews Falsetta from Pattern Bioscience, and the poster was titled Bypass Culture with Confidence, Multi-Site Reproducibility of Phenotypic Polymicrobial Microorganism Identification and Antimicrobial Susceptibility Testing with the Pattern Pneumonia ID AST panel. And the study used over 500 bronchoalveolar lavage and endotracheal aspirate specimens, and the study showed a very high percentage of reproducibility for organism identification and susceptibility testing across polymicrobial targets, supporting this direct-from-specimen phenotypic platform as a realistic way to bypass conventional culture and shorten time to an actionable AST result in pneumonia. And the next poster was from Christina Kosiansky from Mayo Clinic, and it was about phage susceptibility testing in Pseudomonas. And in this study, they compared plaque-based and liquid-based phage susceptibility testing for Pseudomonas aeruginosa isolates from a randomized clinical trial in cystic fibrosis. And they asked which assay format best predicts how a multi-phage cocktail will actually perform in patients and show high overall agreement between the methods, while also identifying where they diverge. The 4-phage cocktail provided broader coverage of Pseudomonas aeruginosa from cystic fibrosis trial participants than any single phage using either plaque assay or liquid assay. Okay, so the next poster was from Jasmine Benbey from the Walter Reed Army Institute of Research. And this poster was titled Two Phages Active Against XDR Acinetobacter baumannii. Clinical isolates represent novel taxa. And in this poster, her team described 2 newly characterized phages, EAB1 and EAB9, that are genomically distinct from known phages and show lytic activity against multiple extensively drug-resistant Acinetobacter baumannii isolates in a 106 strain diversity panel, making them promising candidates for future phage therapy against this critical priority pathogen. Then the next poster was from Daniel Navas from Advent Health Orlando, and this poster was titled Rapid and Routine 24-Hour Antimicrobial Resistance Report for Mycobacterium abscessus group by whole genome sequencing. And in this study, they did sequencing on 20 Mycobacterium abscessus isolates, and it showed that genomic resistance predictions for key markers like ERM41, RRS, and MAPTET-X had a high categorical agreement with extended phenotypic susceptibility testing. Because routine susceptibility testing for Mycobacterium abscessus can take a long time, this supports whole genome sequencing as a way to generate 24-hour resistance report and help guide therapy for this organism. And then the last poster, and certainly not least, as I said earlier, there was a lot of great content out there. But the last poster that I'm highlighting, it's from Tria Bat from the Broad Institute, and they presented BATLOG, which is bacterial and AMR detection by Sherlock, and it's a rapid, low-cost CRISPR-Cas13a diagnostic that detects 9 bacterial species and 4 resistant genes directly from positive blood cultures. And they validated it across more than 2,000 individual reactions and reported a very high percent reaction level accuracy with both fluorescent and lateral flow readouts, positioning BATLOG as a practical, scalable solution for pathogen and resistance detection in resource limited settings. So, shout out to everyone, such great work. Thank you for everything you do up there. And I enjoy all these presentations and all these posters. So, thank you. Okay, so to kind of wrap it up, let me tell you what I saw on the vendor side of things. And just like on the posters, the overall theme it's rapid susceptibility testing and gene sequencing. So that's the overall prevailing theme. But as I was walking around, you know that if you work in the lab, sometimes you know to help with the high volume, you have this automated systems that do streaking. For example, you have like the Keystra, you have the Wasp. So I saw one from Center Diagnostics, which is distributed by Acer Scientific, and that is the Autoplaque system. And this is an automatic plate streaking and labeling system designed to standardize inoculation and help with workload. And tools like this, they help labs that have like huge volumes and they can free up techs to work on more complex tasks. Okay, and then vendor related, I went to a talk from Copan that covered their automation system. They talk about the WASP, and they also talk about Phenomatrix and Colibri. And WASP is their instrument for automated plate streaking and specimen processing. Phenomatrix is the AI layer that reads digital images of plates and applies rules to flag plates as no significant growth, mixed flora, or presumptive positives. And then Colibri takes it further by automatically picking colonies and preparing standardized suspensions for downstream testing like Molotov and susceptibility testing. And this is very neat. And even in the talk, there was a microbiologist from Canada, and he was talking about how they use Phenomatrix over there for some chrome agar plates. So it is very interesting to see the direction that the lab is going. We're seeing all this automation. Now we're bringing AI. So these are very interesting times. Well, friends, as I said, this is not a comprehensive recap, right? So I always share the talks that I enjoyed. I highlight some posters, but as I said earlier, there is so much content and so many wonderful presentations and projects that if I was to mention everything, it will be a very long episode. So shout out to everyone for their great work and shout out to ASM for a great meeting. Well, friends, there you have it. That was the episode. I hope you enjoyed it. As always, I enjoy sharing this information with you. And thank you so much for the support. As usual, you can find us on any podcast platform. So if you go to Apple, Spotify, Amazon Music, GoodPods, we are right there. So please subscribe to the podcast, download episodes, and if the app allows you to do so, please leave a review. As far as socials, you can find us on Instagram, TikTok, YouTube, Blue Sky Social as Let's Talk Micro, X as Let's Talk Micro 1, LinkedIn as Luis Plaza. And then there's the website, which is letstalkmicro.com, and an email address, which is letstalkmicro@outlook.com. In the website, you can find information about licensing, study tools, episode updates, and more. So either via the website, email, or social media, you can provide feedback. You can suggest any topics. So I look forward to hearing from you. And if you would like to support us in any way, you can do so via Venmo or via Buy Me a Coffee, KOFI, both links available on the show notes. Okay, my good micro friends, you know that if you're in this community, whether microbiology, infectious diseases, you know that learning about antibiotics can be very hard. So, if you would like to learn more about antibiotics, please check out www.learnantibiotics.com and the Learn Antibiotics book available on Amazon. These resources include cheat sheets, practice tests, games, and more, and they are being used by thousands of people worldwide and may be helpful for you or your colleagues. And as always, these are some great resources from Dr. Timothy Gauthier, a very passionate pharmacist. And the Let's Talk Micro podcast is supported by the American Society for Microbiology. To learn more about microbes, visit asm.org. And that, micro friends, is the end of this episode. I hope you enjoyed this recap for ASM Microbe 2026. As always, I enjoy sharing this information with you. And stay tuned, the live episode will be released soon. So thank you for the support. Great things coming your way. And please continue bringing that passion to what you do. It's so important. You do such great work. So, as always, stay motivated, stay safe, and of course, continue talking micro. Until the next time. Bye.